My name is Whitney Stevens-Sostre, and today I'm going to talk to you about part of my thesis work, which is understanding ion channels, our electrical signaling machines. And so I specifically work with the EAG1 voltage-gated potassium ion channel. Ion channels are proteins that are embedded into the membranes of our cells, and they allow for the conduction of different ions. Voltage-gated channels are also ion channels that open and close in response to changes in membrane potential. And here I'm showing you a structure of the EAG1 voltage-gated potassium channel that I study with calmodulin. That is another protein that binds to the channel to regulate its opening and closing. There are many disease-associated mutations in EAG1 channels, which include epilepsy, Temple-Breitzer syndrome, Zimmermann-Leban syndrome, and also cancer. And so understanding how this channel works, we are also able to better understand how this channel's dysfunction contributes to these different diseases. And so why do ion channel mutations cause so many diseases? This is a really big question that is addressed by different researchers. And in our case, we really hope to understand how the ion channel works normally to better understand how its dysfunction can contribute to different diseases. And so to do this, I use structure-function relationships, a structure-function approach. And this is reflected by this quote by the famous physicist Richard Feynman, who said, "It is very easy to answer many of these fundamental biological questions. You just have to look at the thing." And so I look at the structure of the EAG channel, and I am trying to understand how calmodulin binding to this channel inhibits its ion channel currents. To do this, I first look at the channel. I make changes to this ion channel structure by introducing different mutations, and then I test how this works using electrophysiology. Specifically, I use 2-electrode voltage clamp to measure EAG1 currents. I take Xenobus laevis frogs, extract their frog eggs or oocytes, and then I express the EAG channel in its normal and also mutated versions into these different individual cells. Then I use 2-electrode voltage clamp, and I introduce voltage changes to these cells that then generate the different EAG1 currents. And so by understanding how this channel works normally, we are better able to understand how its dysfunction can cause the different diseases I mentioned. Thank you.